thank you. It's a real pleasure to be here, and actually it's also a great honour to be on the same stage as Professor Jarvis. When I joined this journey on tobacco harm reduction in the 80s, he was a guiding light there, and he remains a guiding light. And when I've got a dilemma on nicotine, what I think is, what would Martin do? So thank you, Martin, for everything you've done. I should read my conflict of interest. I work for British Men Tobacco, which is a tobacco and nicotine company, and the research I'm going to show you was either funded by us but done by independent labs, or was done in our laboratories in Southampton in the south of the UK. Okay, I'm going to start with the risk continuum. And Ian Jones earlier today showed a risk cliff. It was interesting. It was a phrase I coined in an article for the Food, Drug and Law Institute, and was very glad to see Ian using it. I think we have to be very careful on chemistry, on thinking that we can define what products are on a risk continuum on the basis of chemistry. And I'm starting to see claims of this product's got 90% or 95% or 99% less toxicants. They may all be the same biologically. So the scientific assessment that needs to be done for these tests is much more than a simple toxicant evaluation. And ultimately, it needs to be epidemiology. So we know how dangerous cigarettes are because we've had epidemiology now for many, many decades showing they're extremely risky in terms of lung cancer, chronic obstructive pulmonary disease, heart diseases, many, many other diseases. We know nicotine is pretty benign on the basis of use, particularly through medical devices. So that sets two ends of the risk continuum. We know snus, because of its use particularly in Sweden, but also oral tobaccos in the US, are substantially less risky, probably around the 95% less risky than cigarettes. E-cigarettes are still evolving. We have no long-term epidemiology. The way, again, in that article I described where Public Health England and Royal College of Physicians and the Department of Health in the UK and now some other governments, including Health Canada, have come out, is educated conjecture. And it's based on good science, so it's highly educated conjecture, but we can't absolutely nail where vapor products are on this risk continuum, but they are very likely to be in that kind of 95%. Tobacco heating products are even harder. They're even less science than one. That science tends to have come out of the tobacco companies' laboratories. Not that that's not good science, but independent verification is going to be really important. So placing these is going to be more difficult. So we're trying to do the science. We have a duty to our consumers to make sure we understand these novel products as well as anyone does. And also we're trying to communicate that science to inform the public debate on what should be done and are these part of a tobacco harm reduction story. It's worth showing the quotes from Public Health England and Action on Smoking Health. In the UK, I'm very fortunate to live in the UK. I'm travelling a lot these days to various countries. And in the UK, I had a presentation to our Parliamentary Select Committee on Science and Technology, where the world has completely changed. The parliamentarians weren't seeing tobacco companies in front of them, they were seeing makers of electronic cigarettes, which they think is going to have a great benefit to public health in the UK and will accelerate the reduction of smoking incidence in the UK. I was also recently at a presentation in Brazil, in their regulator Anvisa with the WHO, who banned all these products completely. So the environment is very different. I think the UK is a really guiding star for this, but it's going to need to be more and more science on these products to convince other parts of the world that this is a viable way forward. The manufacturer, I think, has a particular dilemma, because we are trying to not improve these products, so we need to understand these products really well. We can't say they are less risky definitively because we don't have that long-term science, so we're in a place where we have to communicate honestly and truthfully to consumers and to regulators, whilst at the same time saying, well, this is where there's still uncertainties in the science. The large reductions in toxicants may or may not translate to reductions in risk. We do not know the dose-response curves for all the individual toxins that are present in cigarette smoke. So you have to be very cautious about a simple toxicant reduction claim. What is, I think, going to be great advantage is the development of product standards. I know there's work in ISO and other places on e-cigarettes. That's going to help both consumers understand that these are been governed to a certain level of safety, and helps, I think, push the category into even lower risk territory. And when you do this science, you've also got to figure out ways that product innovation, because this is disruption. And it's disruption that will come from all parts, and small companies, big companies. And you have to be able to account for that disruption still being part of this tobacco harm reduction goal. So our science has to be multidisciplinary. You can't just do chemistry. You have to do chemistry and toxicology and biology and clinical studies and population studies, all put together to try and come up with a picture. So what are these products doing for individual risk and for population risk? In theory, there's no reason we can't make e-cigarettes virtually risk-free, because it's a very simple system. And as long as there's no issue with the continued exposure to propylene glycol, glycerol, maybe whatever other carriers we have in the future, maybe water, and we don't put any toxicants in, and we don't let the glycerol and the propylene glycol burn at the coil, then we should be almost toxicant free. And then that's a lot easier conversation to have with the regulators. They say, well, it's not 95% or 99%, it's zero. It's zero, guys. So therefore, you're likely, very likely, to come forward with positive health consequences. This chart, just to explain it, so the target on this side with all the chemicals on it is all the harmful and potentially harmful toxicants that are found in cigarette smoke or tobacco defined by the FDA. And the various bullets are either Health Canada or the World Health Organization's target toxins reduction. When you go to Vape pen, in this case, you go to vapor, most of them aren't there. Okay, so absence of a toxicant is a much easier thing to explain to people. It's just not there. It's there in smoke, it's not there in vapor. The ones that are there in green are over 90% reduced. So really, just from looking at the chemistry, I've been going on about, well, you can't just use chemistry, but looking at the chemistry shows great promise in a good vapor product. And those reductions will vary depending on whether you, what those lists, if you look at the middle list, you're usually down about 99. If you look at the big list, you're down at 92, which again is why it's quite tricky to try and make a claim for a particular product against all lists. But biology really needs to kind of kick into this. So the consequence of that changed chemistry in the vapor product is you go from cigarette smoke, which is highly mutagenic, so it's causing damages to cells which could cause cancer, not necessarily always, to not at all mutagenic. And as a scientist, you hate to see a zero result. You're kind of thinking, okay, so it's a flat line, so I don't know whether I haven't dosed the cells properly or I haven't done enough to actually elicit response. So we've pushed and pushed this system to try and get it to at least show a little bit of mutagenicity, but we can't, not with vapor. It's completely flat, even after pouring 900 puffs onto the cells. We are great believers in 21st century toxicology, so developing in laboratory models that mimic key events in a disease progression. So each of those chronic diseases has various states of progression, usually starting with inflammation or oxidative stress, and moves on to more complicated biological processes. So we use human cell lines in our laboratory mimicking those key events, and then we test cigarette smoke versus vapor. For the test for oxidative stress, cardiovascular disease related, genotoxicity, tumor promotion, we don't get any reaction from the Vape E-pen vapor, whereas obviously cigarette smoke triggers off a large response in every case. You move further on into try and look at it holistically in the body and using systems biology, you can map 50,000 genes in a cellular system that are firing, that are upregulating or downregulating. And we have knowledge of which pathways they can lead to diseases. So cigarette smoke, as you can see in that kind of volcano plot on the side, 3alpha-F is the reference cigarette from University of Kentucky, fires off this gene. So genes go freaky when they get smoke on them, and you know that's the starting point for them being damaged, which could lead to diseases. When you do E-pen, and actually what we've done there is E-pen vapor, and then E-pen vapor times 2 nicotine to see whether there's any effect from nicotine, and you really get virtually no changes to the gene regulation or upregulation. Okay, let me move from vapor, which I do believe eventually will be proven to be much less risky than cigarette smoking when we start to do, and we are doing long-term clinical studies and looking at biomarkers of biological effect. Tobacco heating products are trickier. So when I'm talking about absence, you should have an absence of toxins. I think destination, when we get to generation 3, generation 4 e-cigarettes, will be virtually no or no toxicants, just the nicotine. But tobacco heating products are inherently going to have some toxicants because you're using that raw material tobacco, which will always contain some things which will come over to the vapor. I think there's work needed to be done to define just what is a tobacco heating product. It's going to relate to temperature and lack of combustion. So if you're not burning it, you should have a good consequence. If you start to burn it, you're going to have a poorer consequence in terms of toxicants and the biology that happens. We have a kind of 5-step test in our shop which looks at chemistry and physicals to see, okay, is this product something we know is not actually combusting tobacco? That includes just looking at it to see what happens after use, how much of loss have you had. It looks at markers of combustions like carbon monoxide, carbon dioxide, and oxides of nitrogen, and it also looks at those chemistries I showed you before. The regulatory environment of this is developing. So where I think we're getting firmer and firmer from a regulatory point of view on e-cigarettes being substantially less harmful than cigarette smoking, the few people that started to look at tobacco heating products are thinking, yeah, these are probably very likely to be less risky than smoking, but maybe more risky than vaping. So they're kind of in a slight hinterland, and we've got a huge spectrum. So Korea says low-tar cigarettes are better. The Korean FDA, which was a slightly strange finding from the analytical data they collected, which shows significantly lower toxicants, whereas Public Health England, ESA, and actually German Risk Assessment Unit last week say probably quite a lot less risky. The chemistry is good. You can, as long as you keep the temperature down to a level where you're not combusting, get levels of toxins compared to cigarettes substantially reduced in the kind of 95% level, 1995. When you do high throughput screening, so you're looking here at biological endpoints very quickly that go through a variety of things that are bad for cells, bad things to happen to cells, whether that be oxidative stress, whether that be changes in the DNA structure. You see cigarette smoke fires them all off, and the vapor from a tobacco heating product doesn't. We've done a classical way to do this would be an animal study. We don't do that. So we've done a laboratory study which looks at mimicking cessation compared to switching to glow. And you see week 2 to week 4, what happens is when you continue to expose these human cells to smoke, you get a continued inflammation, inflammatory cytokine expression. So the cells are screaming, help, I don't like this, basically. And you do the condition, you do 2 weeks of exposure of these cells to smoke, and then effectively switch them to glow or switch them to air. What you find is a recovery. The cells are getting better after that switch. So the classical design, usually done in humans or animals, but done in this case in our laboratories in Southampton, shows encouragement. Clinical, in the end, is going to be the key to defining this. This is our Japanese clinical study looking at biomarkers of exposure. And it's something that we presented in the posters by Nathan Gale later this week or during this week. And it was a study where we asked volunteers to come to a hospital. One group carries on smoking, one group is asked to switch to glow, and one group is asked to quit for the period of time. And what you find with markers that you know are in cigarette smoke, like carbon monoxide or the metabolite of benzene, stay there. When you get switched to the tobacco heating product, in the case of CO, which get out of the body quite quickly, in a day you're down to where you would be if you quit. With benzene, because benzene takes a longer time to come out of the body, it takes a couple of days to get down, but it tracks cessation. When you look at a broader range of toxins, it's not happening completely to all of them, but a lot of those toxins are kind of tracking the group that was quitting. And the other bit of, when I was talking about the risk continuum, the other bit of epidemiology we know is what happens to people when they quit. The risk for heart disease goes down reasonably rapidly. The risk for lung cancer, depending where they start, goes down slower, but we know they recover, which was also that reason for doing that laboratory study on cessation. Okay, so that's it. Rapid tour of my world of science. We try and publish everything we do. We try and publish it in open access journals so everyone can see that science. And we're trying now to try and also put it together, because it's a multidisciplinary science program that has to be mushed into one story that makes some sense for people. So we've just launched, really thanks to Sarah Cooney in my shop, the R&D science report for this year. It features the products. It features our new vaping product, which will have a very clean vapor because of the way we're generating it, not with a coil and wick, but with another system. It looks at our smokeless innovations. It's looking at our tobacco heating products. So do go see the store. These are downloadable both on Android and on Apple. That was my advert. I'm not for free, so I'm not going to make any money out of that. Okay, so summary. I think the data, particularly on vapor, that we are generating is consistent. We need to do more, and actually Public Health England and Royal College of Physicians report said, look, this is where we think it is, but there still needs to be more and longer term research on these products and product standards. So we're doing a year-long study looking at biomarkers of biological effect, both for tobacco heating products and for our next generation vapor product. We need to get more independent research done on tobacco heating products. That's hard because we can't fund that typically, can't in the UK, and if it is funded by us, it's not seen as independent, but we need to encourage or find methodologies where either we give products to academics or other ways in which the research could be done on them. And that was what I was going to say. Thank you.